joining us in this compost tea talk. This is part of an on-farm community-oriented composting project that the Composting Association of Vermont is leading, and it's funded by USDA Rural Utility Services. I'm Natasha Duarte. I'm the director of the Composting Association of Vermont and lead on this project. Also on this call are Athena Lee Bradley and Kat Buxton, my co-trainers for this project. The goal of this project overall is to develop sustainable community-oriented composting sites with farmer partners to manage food scraps, farm residues, manures, and other organics in a responsible, closed, nutrient loop way. This is today's compost tea talk, and I put that in finger quotes and quotes on my slide because some people think that we're going to be talking all about compost tea, which we have done in the past a little bit, but this is just our take on like a lunch and learn or a coffee talk kind of thing. We are joined today by Lynn Fang from LA Compost. Her talk is titled As Above, So Below, a Whole Systems Perspective on Soil Ecology. Lynn is with LA Compost now, but she received her master's in plant and soil science from the University of Vermont and specializes in soil and compost ecology for composting, regenerative farming, bioremediation, and native habitat restoration projects, really integrating that biological, chemical, and ecological indicators in this holistic view of soil health and what soil health means. Lynn is going to be sharing her perspective on how above ground design and management practices influence soil health structure and microbial ecology and vice versa. I just wanted to let folks know that we're going to go through the presentation first and then have a Q&A discussion session afterwards. But there's no need to wait to put your questions into chat. I will be monitoring the chat box. And when we move into that part of today's session, we'll start with those questions. And if we're a small enough group, we can open up our mics and have a good conversation as well. Lynn, with that, I just invite you to take it away.
Thanks so much, Natasha. Thanks everyone for having me here today. As Natasha mentioned, I work for LA Compost and I've been growing a soil microscopy program there where we offer one-on-one qualitative assessments on microbial ecology. So people can look at their soil and compost samples under the microscope. And we're building that out into educational programming. We offer community workshops where we bring the microscope to different communities where they might not have access to this kind of information and share that knowledge with those folks as well. Today, I'm going to talk about soil ecology in the context of a whole systems perspective. And I like to think about that as how the above ground ecology influences the below ground ecology and vice versa. So the parts that we can manage are above ground more directly, thinking about the design of the land, ecological design, as well as land management practices. All of those things influence our soil health, and that can vary from organic matter content to mineralogy to soil microbiome. And so all of our choices that we make in our design and management above ground influences these soil health properties, especially influences the microbial community that's present and the various groups that might show up, become more dominant, and can also influence other things like how much soil carbon can be sequestered, how much nutrient storage is available, which is also related to the biology. So organic matter is really essential to a lot of the things that we value in soil health. And that is only a small piece of the pie, like maybe about 5 to 10% of soil itself. And that consists of not just the biology, but also like decaying litter, plant roots, as well as stabilized organic matter like humus. And supports structure. It can really strengthen that to resist erosion. So that's related to what we call aggregate stability and how strong and stable those soil aggregates are. And also helps to improve nutrient availability. Our microbes help us to cycle nutrients as well as store them. And our organic matter also helps us to store more nutrients, the humus kind of particles. And this organic matter also helps us to sequester heavy metals, remediate pollutants. The structure of these aggregates helps us to infiltrate and retain more water. So it helps us to reduce our need and demand for irrigation, and as well as sequester more carbon. There's a really great soil scientist named Ratan Lal who's done a lot of great work on soil carbon, especially. And so he really emphasizes these above ground practices that really help to increase our soil carbon pool. These are practices of returning organic matter to the soil, so mulching, taking your plant trimmings, your wood chips, and spreading them over the soil. And that helps to retain water, build biology, and build structure. Conservation or no tillage. So this is really going to reduce the disturbance of the biology and the soil structure. So, I mean, you can imagine if there was a giant rototiller that was ripping up our homes and streets and things like that, how much energy it would take to try to rebuild that. So that's kind of a similar concept of what's happening in the soil. We have these strong aggregates with their own microbiomes in them, fungal networks spread throughout the soil that's kind of holding it together, root networks that are holding it together. So the tillage is really going to rip that all up and take more energy to rebuild that structure. Cover crops really help to support microbial life and especially nitrogen fixation into the soil. So they really help to build soil. They also support more organic matter formation. And our kind of holistic nutrient management includes compost. So compost plays a critical role in soil fertility and also maintaining that organic matter. We can also use manure and rotational grazing as part of your integrated nutrient management system. And integrating water conservation and harvesting features, especially in more arid areas. So that's like looking at infiltration channels, swales, things like that to kind of intentionally divert water to different areas so that can be captured and also provide water to plants around those areas. Something I have come across consistently is that more diverse cropping systems really improve soil health and soil carbon, such as agroforestry that can be integrated, with trees, livestock, crops, etc., and like rotational grazing, like all kind of integrated together. So more above ground biodiversity improves below ground diversity, which is one of the principles to suppressing pathogens and disease. That ecological redundancy in the biodiversity ensures that you have more nutrient availability, more resiliency to climate change, things like that. This is another interesting study done by Ernesto Mendez, actually at University of Vermont. And this is looking at how cooperative management approaches influence coffee agroforestry and soil health. So this is looking at more of the human kind of dimension, looking at our social organization and decision-making process. So it looked at two kind of cooperative coffee farms. One cooperative was more commercially oriented and more interested in coffee as a cash crop and making more money. And the other cooperative was interested in more tree biodiversity conservation. So they were more biodiversity focused, maybe a little bit more focused on ecology. They found that both of these projects had no change in soil carbon content, but there was increased species richness correlated with higher levels of cation exchange capacity, which is related to nutrient storage and other important nutrients, with the cooperative that was more focused on tree biodiversity conservation, which is really interesting. So the soil microbiome itself is a small slice of the organic matter content of soil. So it's like 0.5% of the soil itself, but it drives so many of the critical ecosystem services that we really need and value in soils, including climate regulation, carbon sequestration, and water purification, nutrient availability, habitat for microorganisms, also watershed management, obviously providing our food, fiber, and fuel. But they also are a source of pharmaceuticals and genetic resources. Antibiotics have been produced that were discovered from soil microbiome. And soil microbes are able to adapt because they're exchanging genetic information very quickly, and they're able to learn and create new compounds more quickly. So they're a source of antibiotics, antifungals, or pathogen kind of resistance compounds can be created and discovered in soil microbes. Of course, as well as the foundation of our built environment and cultural heritage and history. So I wanted to touch on some of the functions of soil and human microbiome that is very similar. So we also have a microbiome in our body, and it is the gut microbiome that's very powerful, but there's a lot of microbes in our body in itself. And I've also read that a lot of our genes can be traced back to bacterial genes. So we have this evolutionary relationship with microbes. But they help us to enhance our metabolic pathways as they do in soil as well. So help to break things down, enhance nutrient availability. And the biodiversity and biocontrol organisms help us to suppress pathogens, improve our immune response. They can enhance our growth and influence our behavior. Microbes in our gut can produce precursors to neurotransmitters like dopamine, and that helps us to modulate like stress for us. And then for plants and soil, it helps to help them adapt to their stress, which is more like climate resiliency and also like immune function. And so the creatures in our soil microbiome consists of our primary decomposers, bacteria and fungi, which are feeding more directly on decaying litter, and higher level organisms such as protozoa, which feed more on bacteria, nematodes, which can feed on wide array of organisms, including other nematodes. And then we get into our microarthropods, which can feed on lower order organisms. And there's also other organisms on that mesofauna level, such as tardigrades, rotifers, which can also feed on all of the little guys. Tardigrades also can feed on algae as well. And rotifers also feed on all of these little guys. And then we get into our macrofauna, like insects and small animals, worms, things like that. And so all of this activity, this feeding and metabolism, these higher order organisms like protozoa and nematodes, they're going to release nitrogen because there's more nitrogen in the bacteria and fungi that they're feeding on. So they excrete that, and that is in plant available forms. So that's stuff that plants can take up. They can also secrete growth promoting hormones for plants. So that's something that's been documented in protozoa. And so plants can benefit from that. So there's a lot of beneficial compounds that come out of the soil food web metabolic activity, and that fuels our climate regulation, our nutrient cycling, plant fertility, things like that. It's just an example. I think it's called the microbial loop hypothesis that's been documented, where the plant root exudates attract bacteria. And so the root releases some sugars, and those we call exudates, so you attract bacteria. And the protozoa are attracted to the bacteria, they feed on that, and they release nitrogen and so it's just kind of a little breakdown of what it means to see these different microbial groups when you're looking at them under the microscope. So the bacteria are primary decomposers. They can also be involved with fermentation, biocontrol, which is like suppressing disease organism. So these are going to be prevalent everywhere, even in kind of disturbed ecosystems. So they have higher turnover and adaptability. Fungi are also primary decomposers, but they specialize a little bit more in woody material and the tough kind of plant cell wall material. And they're really critical to forming humus or stabilized soil organic matter. And they tend to be more efficient in carbon sequestration. And I feel like they're also more important to pathogen suppression as well. So they're going to be an indicator of that presence of more woody material, and that you're going to be creating more of this stabilized organic matter in the soil. And so they're going to be an indicator of a more stabilized, mature, more perennial shifting kind of ecosystem when we're talking about like ecological succession. And then protozoa, that's an indication of an abundance of bacteria, and that there's kind of good nitrogen nutrient cycling happening. Nematodes, same thing. Also, there's an abundance of nutrients and organisms that they can feed on, and they're also contributing to nutrient cycling. And they've generally been considered a sign and a tool for assessing ecosystem maturity and stability. But I have to say, I see a lot of nematodes in more immature compost too, and their population levels also increase with moisture. So it's not just the abundance of nematodes, but it's the particular feeding type. So we distinguish them between bacterial, fungal feeding, plant root feeding, predatory, or omnivores. And so the presence of more fungal or omnivore feeding nematodes is going to be your indicator of a more perennial, mature, shifting ecosystem. Whereas if there's a lot of bacterial feeders, it doesn't necessarily mean it's like a mature ecosystem. And then we have our microarthropods, which we also sometimes see in the microscope. And these can be shredding particles to make them smaller, and they're also feeding on smaller organisms and supporting organic matter formation. Some of the common patterns we'll see in different samples. So I'm looking at a lot of urban kind of soils. So a lot of people are like, oh, I just moved into a new home and soil has been vacant or abandoned. So usually we see just a lot of minerals there. There's probably some bacteria, like it could be low to moderate population abundance of bacteria. And there might be a few little bits of fungi, but that's it. There's really low microbial diversity and abundance, and we don't really see a lot of organic matter aggregates or particles there. If we look at more of, for example, a garden soil that's been well amended with compost, organic matter, then there's a lot higher microbial abundance and biodiversity. So we see good representation from bacteria, protozoa, fungi, potentially even nematodes, and there's more organic matter particles. So that's kind of our indication. Also in compost, when we have a fresh hot compost pile, it's going to be teeming with microbial life. So the most abundant microbes is going to be in this fresh hot compost. So that's like after you've built your hot compost pile, it's been like a week or two, this is the most abundant time. So lots of bacteria, amoeba, there could be pathogens too, because that fresh food waste is still decomposing. There could be a lot of lactobacillus because there's that food waste fermenting in there, and a lot of bacterial feeding nematodes is also possible to see that. In more mature compost that's cooled down, it's more aged, it's created its soil structure, soil aggregates. It's higher on fungi, and we can see when we see nematodes, they're more like omnivores or fungal feeding nematodes. And so the microbial population is abundant, but it's not as abundant as the fresh hot compost, and it's a little bit more moderate. So that's what you would expect to see. You want to see that. And there's also going to be more we're pretty much done creating a little soil microbial ecology pamphlet to help people understand how to interpret their microbiology. And so this is about to get published and be freely available on our website. We have like a compost guide on our website. It's a trifold pamphlet that's freely available for people to use as reference. So this is something that we're going to be publishing very soon. So this is like a bifold pamphlet. So the front page is on the right, and then I think the last page is the ecological succession page. So that's where we're looking at that above ground and below ground ecological succession, and how we can interpret that from what we see under the microscope. And our graphic of soil ecosystem services that we adapted for what LA Compost is doing. So we include our community well-being in there, people that we work with. And we adapted the soil food web to kind of a compost food web, which is based on what we see in the microscope and also in the compost pile as well. And the bottom has our more specific microbes as my introduction of what they might look like and what they mean. So this is a very brief or very intro level to understanding your soil microbial ecology. Okay, so now I'm going to look at some photos, and then we can go into questions. And I know people also sent some photos and videos, so we can talk about those. But this is what a nematode looks like. Its mouth is kind of in this organic matter particle, so you can't really see, what that mouth shape is. That's generally how we identify, at least based on feeding group, the mouth structure. The little tiny circles are bacteria, so there's a bunch of them around. There's also little funky clumps of things. Those are probably more like like minerals. They can also be crystal shaped, but sometimes they're in these little clumps too. And that on the top right, it's this fuzzy, large organic brown thing. That's the organic matter particle. So this one is probably more clear, these dark brown clumps that are really large. Those are organic matter particles. They can vary from fully decomposed, partially decomposed materials. You'll see kind of clear crystally things. Those are going to be minerals or salts. This is a nematode in the center here. Also, the mouth is fuzzy, so it's a little bit hard to tell. This one below it is kind of like this oval egg shape. So it looks like it could be like a testate amoeba, which is very common, oval shaped amoeba. It's a good sign of abundant organic matter. So it's very common. So it's good to see. So there's a lot of activity here. There's a lot of bacteria. We have our amoeba, a nematode, lots of organic matter. So this is a rich substrate. It's probably a compost or a highly amended soil. So that's what that looks like. But I've been fortunate to see like 2 tardigrades in compost samples before. So that was really exciting. Tardigrades are their own phylum in itself, and they're the most resilient microbe. They can survive deep space. They can go dormant and then wake up on another planet and start life anew or something. I don't know. I like to think about that. But this one has eggs inside of it. It's like molting or about to give birth. So that's what's happening in this picture. And this is from somebody's home compost pile. So you see there's also a lot of bacteria happening. There's a lot of abundance, microbial abundance. This long clear strand is actin. So that's what that looks like. And you can see there, it looks like there's maybe little circles inside, and that's kind of the structure. So it's like a bacteria, it's a circle, but they link together to form long filaments. They're also very ubiquitous in soil and compost, so they're a good sign to see. They're good decomposers. They're dominant in the hot compost phase, and they give soil that like sweet earthy smell. So we can see that oftentimes there's a lot of actinobacteria in there. This oval-shaped guy is a testate amoeba, so that's very common as well. And there's this like skinny little rod here. That's our lactobacillus, and there's maybe another faint one on the right side as well. And those are fermentation organisms, so it can be a sign that kind of activity is happening. They do live in anaerobic conditions, but I think they're also facultative, like aerobe, so they can live in oxygenated environments as well. So they can kind of move between both states. This large round circle is probably some kind of an amoeba. There's also circular forms. There's one called like Cercozoa. It's like a circular form amoeba. So that's what's happening here. This is kind of a example of those bare soils. So this is just like a lot of minerals, and then there's a few bacteria in between them. So not quite as much going on here. This is a pathogenic bacteria. It's spiral shaped. So that's the spirochete, and it's not necessarily going to give you an infection, but I often see this in like fresh manure, fresh compost, anaerobic soils, immature compost. Some of the industrial composts around here anyways, usually immature and hot, and they'll have these pathogens in them. So it's just usually an indicator that compost is not finished, not mature, or something's a bit off about the soil. So it can use it as an indicator like that. So you won't necessarily get sick. I guess if you're like really stressed and like you're, you know, doing a lot of work by a bunch of manure, by some pigs, a pigsty or something, like you might get sick. But just like seeing it under the microscope doesn't mean you're gonna get sick. But yeah, it does cause like intestinal kind of infection. So this is also a fresh hot compost. There's a dividing amoeba and some lactobacillus, just a lot of bacteria in there. So it's just this overwhelming kind of activity in that hot compost pile. Same thing here, a lot of activity. And yeah, so that's my introduction. Do we want to go into questions, or we could look at some of the photos that people sent.
Yeah, so thank you so much. And I apologize, I turned my camera off just because I was having an unstable internet connection warning come up. So I'm not going to share my camera again, but I'm here. And we have a couple questions, and then do you have the other photos? I think it would be great to look at some of the photos, and I can pull them up or you can pull them up, whatever is most convenient.
Yeah, I have them, so I could share that screen.
Sure, that'd be great. And I don't know how you feel about me asking you a few questions while you're bringing that up, or do you want to just get it up and then we can
Sure, yeah, you can ask.
Okay. So one question that we got is the pictures you've been showing us show, well, except for the more degraded soil, show a lot of life, but really the reality is that you're taking a sample and then you're taking a drop of water with a sample in it and looking at it. If you're looking at a sample and you don't see a lot, do you feel like that's indicative of a lack of diversity? Or is there a minimum number of samples you need to look at to feel that you're getting a good representation of the biology that's actually potentially there?
I think it is good to look at a
Mhm.
couple samples, but oftentimes like they'll look very similar. So the way that the sample is prepared is I'm basically just taking like 1 milliliter of like soil or compost and then
Mhm.
diluting it 1 to 5 with
Mhm.
distilled water and shake that up a little bit, and I just take like basically just like 1 drop of that solution and look at it on the slide. So what you're seeing is like the content that is more easily going into like a water solution. So I've also, people have given me like little bits of wood chip with like mycelium on it, and I tried the little to try to do like the water thing, but I don't think that works so well. And I've also seen people take little slices of, mycelium and like put them on a slide. And that's probably a better way to look at some of these larger kind of things rather than putting them into a liquid solution. So that's something to keep in mind. You're not looking at some of these larger particles or the larger masses of things that have like grown on these larger particles, like larger masses of fungi or things like that.
Mhm.
So that's maybe a little bit of a limitation just in this kind of preparation process. Um, so it's something to keep in mind. And sometimes I do, I look at a compost where I would expect to see a lot of biology, oftentimes like a worm compost and or even just like a food waste compost, but then I don't see a lot of biology. And I don't know, I'm kind of curious about what's happening there too. I think something is not kind of translating into the preparation process
Mhm.
for the microscope analysis. I am sure there's still life in that sample, but for some reason it's not getting into the solution and showing up on the microscope. So I do come across that. I don't think it means your soil or compost is lifeless per se, but there's maybe, I don't know, maybe it's low. Sometimes samples are dry too, especially out here, things dry out a lot. So that can also reduce the biology that shows up. It doesn't mean there's nothing there, but I don't know, it could be an indication that it's low, but it also doesn't necessarily mean your plants are not going to grow well in it because there could be other compounds in there that are supporting plant growth.
Sure.
Um,
Quick, just clarifying question. Can spring water be okay in preparing the samples as well, as long as it's untreated water? So not like a city treated water, but just straight from a spring or a well?
Yeah, it could be. I guess that I was just trying to reduce any variability or influence from the water. So,
Mm-hmm.
just minimizing the biology that's coming from the water. So, I mean, what you could do is just look at the water itself under the microscope and see if there's anything coming up there. And, if there's a lot of stuff in there, then you'll know that it's adding a lot of noise to your
Mm-hmm.
sample. And so then maybe you want to use a more purified water to look at.
Yeah. And that's a good point. And just to remind folks that this is also like a snapshot in time. And so I've had some people give me samples that were somewhat dry, like they seemed like a normal moisture, but it wasn't really what I was expecting. But they had actually collected the samples and dried them, but it was from a supersaturated, it was from a total waterlogged sample. And I think that also can impact the biology that you see there compared to what you might see if if it was less of a moist sample, because it's, if it's wet enough, long enough to actually go from aerobic to anaerobic, you change the complex of biology, right? So just to remind folks that it's a snapshot in time. So we have one question that came in early, and maybe we'll just get to that and then we can look at some of these other fun images. So one question that came in early on was, in your presentation, was how can we make raised beds that help us not drown with all these rains and still do as close to no-till to have better soil health? Like how do you square those two goals?
That's a good till that it won't infiltrate the water as well. Is that right? Because we don't get a lot of water out here, so I don't think about that as much.
Yeah. So we've
mhm,
been flooding here in Vermont for the last
mhm.
month or so. So I think it's more like if you're creating like an raised bed, clearly you're creating that soil substrate that you're going to plant in, right? Because it's now, even if it's on the ground, you're adding material up above.
Yeah.
And so
Well,
does that impact the soil health? Because it's essentially, it's no longer a no-till kind of or low-till kind of
because of all
setting.
of the rain and
Well, and
water.
because you're creating that bed to even
Okay.
be above the water level to plant into, I guess.
Right. I guess like I like to use kind of a sheet mulching process for building soil in a raised bed too, or even kind of a hugel kind of method. So like layering wood chips, burying logs with soil, compost, and then topping it with a good compost or soil blend that can be kind of used right away. And then that bottom stuff will decompose and build soil over time. And so those kind of materials are going to encourage more fungi to develop. Thought is that it will create a more fungal dominant soil substrate, and that will help to create stronger soil aggregate and structure to improve infiltration. I think maybe you're more concerned about drainage, so making sure the water doesn't. You're probably concerned about water logging, so maybe thinking about having drainage at the bottom of the bed, maybe even putting in some gravel at the bottom. And I would still do a no-till or low-till kind of practice in that situation. So you build up your soil substrate in the raised bed, and then you're growing vegetables and just amending, at the top with compost when you're putting in new plants. I feel that should be fine. I don't know if there was a concern of soil compaction or waterlogging with that method, but that's probably what I would do.
Yeah, and I'll just add that Michael, who asked the question, and feel free to chime in if we're missing some important piece of your question, but it's the bare soil rule still comes into play, I think, even in raised beds. So you don't want to have a lot of bare soil, right? You want to keep it covered, and that helps break up the force of raindrop impact as well. And so just because it's, might feel sort of like an artificially created growing area, growing medium that you're making in the raised bed, you still want to follow those same soil health principles of not over-till, like Lynn said, and then also, not having a lot of exposed soil for rainfall to
Yeah,
impact.
it's still good
Mhm.
to have mulch on the soil. And I think if you're concerned about waterlogging, I would try to build it to, yeah, facilitate more drainage at the bottom, gravel at the bottom, like more mulch on top, more chunky stuff in the soil blend, like more wood chippy kind of stuff in there to make sure the soil doesn't get compacted.
Great. All right. Well, thanks for that. Michael's just said thank you in the chat. So I think we covered it. So let's look at some of these samples. These are from one of our partner farms, Firefly Farm at Burke Hollow in Vermont, that Kat Buxton, one of our trainers that works with Ben Tipton at Firefly, took these images.
Great. So this looks like this first photo, yeah, a fungal strand. And the way that we identify it, it's like a long and kind of even, it's like it's just same width all the way through. It's a long strand. There should be these little tick marks to indicate what we call septa. So that's the cell wall for each of the cells. And they can be branching, they can be clear, they can be a little bit brown. I think if the septa are irregularly spaced, that's an indication that it could be more pathogenic. But regularly spaced septa, that's generally an indication of beneficial fungi. So usually what we're seeing under the microscope is going to be the saprophytic decomposer fungi. I don't know if we're really seeing mycorrhizal fungi, because I think there's a different process to look at those. So this is our indicator of decomposer fungi, which is great.
Nice.
So this is a nice long strand. So that's great to see. These longer strands are creating longer networks and stronger soil structure. Sometimes we see little pieces, so that could mean they're more broken up or just maybe not as strong. It looks like this could be fungi potentially at the bottom. Sometimes they do look all crunched up, and they could be in decay as well, like getting fed on by other organisms. I would say since other material, it looks like, kind of various bits of organic matter. And oftentimes we do see the fungal threads weaving through the organic matter particles. Anyway, generally a sign of the soil structure being built. And this one is a rotifer. So it's kind of what it looks like.
Cool.
The head part is at the bottom left, and it has these two like things of like whirling cilia, and it creates these vortexes when it's very active that like sucks in bacteria and protozoa, and that's how it feeds. There are kind of aquatic organisms, so those in the indication of high moisture content, potentially like anaerobic, but I think it's just more associated with moisture. But of course, if you have too much water, it could be anaerobic. But that's what they look like. You can see the internal digestion happening in there. And this is another one of a rotifer. Yeah, it's same organism, so a little bit fuzzy. And let's see what this last one is. So these are moving around. So in our protozoa, we have kind of three different groups of organisms. Flagellates and ciliates really swim around, and flagellates have long tails behind them, and ciliates have tiny hairs all around them. These are moving quite quickly. I think ciliates move a little bit slower. Flagellates move really quickly. And so flagellates are going to have maybe a little bit more teardrop kind of shape for the tail. So I want to say these are probably some kind of a flagellate, just based on the way that they're swimming. And those are great. Ciliates can be more of an indicator of anaerobic conditions. So that's just something to be aware of. If you see a lot of ciliates, it indicates like maybe there's a little bit of an anaerobic pocket that your sample is in.
Cool. And Kat added that the rotifer was from a finishing uncovered pile, and we've gotten a lot of rain. So some of these soils may indeed have some of those anaerobic pockets. Yeah. Or composts. Yeah. Neat. So I think those were the images that we had received in advance. Is there anything else you want to add, Lynn?
I could talk briefly about these keys. So the different shapes of bacteria generally is spherical, round. They can be rod shaped and then spiral or snake shaped. I guess there could be some of these other kidney bean shaped kind of creatures. I don't feel like I noticed those as much, but so mostly what we see are circles, rods, or spirals. And nematodes, this is a key that I reference. So our bacterial feeders have 3 little mounds on them. So if you are able to see a nematode that's not moving around frantically or got its head stuck somewhere, you can kind of see 3 little mounds on the mouth. And the fungal one has this little spike inside. The root one is like round, completely rounded. The predatory one, I think the predatory and omnivore look pretty similar because they have these multi-rounded kind of mouth parts. The omnivore one's more prominent. You can also see inside, so you might be able to see this pincher kind of thing in the predatory one, and then more of a long tubule in the omnivore one. So yeah, sometimes you can spot that. So I feel mostly when I've been able to see the mouth part, it's been bacterial, and then sometimes I omnivore or fungal. Those are the most common ones that I've seen.
Nice.
Yeah.
That's great.
Do you guys have any more questions?
So one question that I received in advance was a little bit general, but we have a little bit of time, and folks who are in the room, feel free to put anything in chat or raise your hand if you want to chime in with your own questions, but I'll go ahead and pose this one while you all are thinking. It was a question, it was pretty general, sort of like, what makes a healthy compost or soil? Is there some level of diversity that you're looking for, either, that you would look at under a microscope and be like, wow, this is a gold standard, I think was their phrase that was used. And I don't know how to answer that. I mean, I think more diversity is good, right? And certainly presence of fungal as well as bacterial, some of the different key players, but I wonder how you would answer that, or if you have a sense of what you get really excited about when you see in a microscope.
Yeah, I think I'm looking for that diversity representation from all the different groups. And then I want to see lots of fungi. I've seen samples, lots of like really long kind of fungi. And I feel like that's great to see. You want to see some nematodes, preferably they're omnivores or fungal feeding nematodes. That's your indication of more maturity and stability. And there should be a good population of amoeba in there too, or protozoa. So generally it's going to be amoeba, I feel like testate amoeba are the most common, but there can also be more amorphously shaped amoeba as well. So I feel like that's what I'm looking for. And then good organic matter particles and clumps in there. That's my gold standard right now. I feel like there's always like some fine tuning that could be done, but I feel like right now that's what my target is to look for that good balance and representation from all those different groups.
Great, thank you. We do have a question in chat. Reed asks, what do you find rotifers anchor themselves to? And is it possible for them to detach, move, and reattach where there seem to be more food available?
And that's interesting. I feel like they move in this, it's like a foot and they like squeeze together and then they move that way. So I usually see them moving around a lot. So, but oftentimes I'll see things stick themselves to an organic matter particles. So most commonly I'll see a nematode stuck on there. I'm not exactly sure how they attach, but there seems to be that affinity for an organic matter particle. And also there tends to be microbes, bacteria around organic matter particles. So I think it's a good feeding zone as well. So that's what I imagine. If they're trying to attach to different things, they're going to hop to different organic matter hotspots.
Cool. Yeah. Any other questions, folks? I was trying to just pull up quickly one more little video that I have. One question while I'm looking for this that I'll ask Lynn. I always get excited when I saw the brochure that you're working on that really explicitly includes community as part of the ecosystem, right? Like here we are, and it's like this reciprocal, we take care of the soil, it increases our well-being. And I just love that piece. And I think it's really spot on for the project that we're working on as well. And in that, I think you've done a lot more, well, clearly you've done a lot more work than I have teaching folks how to use the microscope. And I just wonder if you could talk a little bit about that and how, you know, how people react, or what are some tips for working with folks who may have never looked in a microscope before to get them sort of turned on to this. The video I was looking for is one that I did a number of years ago, and I was tabling at a soil health event, and I brought it and I had it up like on, you know, like on a screen, and I was super jazzed about it. And folks who were there barely looked at it, and I was a little bummed. Of course, I was just totally geeking out on it and so proud that I had caught what I had caught. But anyway, if you could talk a little bit about using microscopes as a tool for community engagement, that would be fabulous.
Yeah, totally. So I talked a little bit about the sample preparation, and I don't know, every time I share this with people, I feel a very common response is like their mind is blown. They're so in awe to see these organisms. I feel like it's a very transformative experience that way. But also, I've taught to grade school students. I did a workshop where it was grade school, middle school, high school students. And the grade school, middle school students were so excited. They had all the questions. Everybody wanted to make a sample and look in the microscope. And then the high school students could care less. They were way too cool to pay attention. And they just wanted to talk to each other and make jokes and stuff. So that was kind of a hard crowd to
Yeah,
please.
yeah.
So I think it depends. I do think young people can be really excited by the microbes. And yeah, generally the adults that I've worked with too, they're usually so into it. And I don't know, it kind of amazes me sometimes. Like I know there's growing interest in like soil biology, but like, I don't know, sometimes I'm like, oh, is this like too hardcore, boring for people? Because sometimes when I'm in a workshop and we're looking at different types of samples, I don't know, it can be a little bit, it takes some time to look through things. And so it can seem a little bit boring. But people who are into it are just totally and so I think there is a lot of interest in this, and there's a lot of excitement for it. So there's a lot of opportunity to kind of, you know, share more awareness around microbial ecology. And getting a microscope set up is, you know, not really a huge barrier. I mean, it's like a little bit of a cost and you have to get some supplies, but it's fairly simple. You know, I guess you need like a little bit of money to get a microscope, but you can get used ones too. You don't have to get the most expensive ones either to see stuff. So I don't know, couple $100 and you can get a microscope set up and start taking a look at what's in your soil and compost, and sharing that with people.
Yeah, that's pretty exciting. Thank you so much. I'll keep trying to persist. So can you all see my screen now? I know we have only a few minutes left, but I thought I'd just throw this up and you can, this is actually not mine. This is from John Culpepper. I couldn't find the one that I was using, but I thought I'd show it in action just because it shows it feeding.
Very nice. It looks like it could be a bacterial feeder.
Yeah, with the two sort of
Little
right
mounds.
there. Yeah. Yeah. Yeah.
Very nice.
Yeah. Anyway, it's very fun. Once I find that once people get started, it's really you just want to keep going, right? And keep exploring and seeing what you can come up with. Thanks once again to Lynn and to everybody for joining us today. Super fun topic, and we hope to see you at future Tea Talks.
Great. Thank you so much.
Thanks, everybody.
Take care, everyone.
Thank you, Lynn.
Bye.
Bye-bye.